My name is Joshua Sampson. I'm 11, and I was wondering, is it possible to make a living creature out of purely stem cells?
I'm Bill Murphy, and I'm a professor at the University of Wisconsin in biomedical engineering and in orthopedics and rehabilitation, and I co-direct the Stem Cell and Regenerative Medicine Center here at Wisconsin. Yeah, stem cells are these special cells inside your body that can multiply indefinitely, so they can make copies of themselves, and they can differentiate. That means they can become every cell type that's present in your body. So generating a living creature requires not just the cells, but also the environment that allows that to happen. And that doesn't happen outside the body. So it requires a womb, it requires a place in which the organism, the new living creature can develop. There are aspects of development of a living creature that we can recreate outside the body, but it doesn't result in creation of a living creature. It results in creation of maybe some specific tissue type or even some specific cell type. And a good example is the tissue chip project that we're working on. So the specifics of that project is that we're trying to generate microscale mimics of the brain. So these are about the size of a pinhead in many cases, very, very small. And they're forming into tissues that are similar to aspects of the human brain. Now, these are not human brains on a chip, but they have similar features. One of the first questions we're asking is, what is the effect of chemicals on the developing human brain? We've been developing these mimics, exposing them to potential toxins and potential non-toxins, and trying to get a sense for whether we can predict whether a certain chemical is going to be toxic to the developing human brain or whether it's not likely to be toxic to the developing human brain. The other areas that outside of toxicology that could be very important and that we're pursuing as well are disease modeling and drug discovery. In disease modeling, we're taking cells that are derived from patients who have neurodevelopmental or neurodegenerative disorders like Alzheimer's disease, autism spectrum related disorders, and we're developing brain mimics from those cells. We can use those now to understand what are the mechanics of those diseases, how do those diseases develop, what are the specific sorts of genes that are upregulated in those diseases, and how can they be treated.